Microbes are responsible for a lot of infections and infectious diseases. And that's one of the reasons that we do study them. When we study them in the respect of how they cause infections and infectious diseases, that's going to be called epidemiology. That's really what we're focusing on in this particular lesson. Now, the microbes around us, they are involved in a lot of different symbiotic relationships. The term symbiosis really means to live together, and there's different types of relationships where the microbes can live with other organisms. One type of relationship is mutualism. This is going to be when both members benefit. We do see this a lot of times between microbes and then also insects. The example that we're seeing in this slide right here is a termite. The termite has a number of microbes in its digestive system that help to digest the wood that the termite is actually eating. It helps it to actually break it down. So that's one example. We do have a lot of microbes that live inside of our bodies, and they do function in a mutualistic way. Commensalism is another type of symbiotic relationship. In this case, one member is going to see a real benefit, and then the other member is not really going to be significantly affected. So that second member will not be really positively or negatively impacted. And then parasitism is going to be when one member benefits and then the other member is actually harmed. So that is what we're looking at when we do talk about infections with regard to microbes. So any parasite that causes a disease is called a pathogen. When we talk about the microbes that are associated with our bodies, we do have what's called the normal microbiota. These are the microbes that colonize the body's surfaces, and they don't actually cause us disease on a normal basis. So those are the normal microbes. These normal microbes, normal microbiota, they fall into two different categories or two different types. The resident microbiota, these are going to be microbes that are established very early during our lives, and they pretty much remain throughout a person's life. These are typically going to be commensal, and so that's that commensalism relationship where one benefits, the other one doesn't really see a real impact. The other type of microbiota is going to be transient microbiota. These tend to remain on the body for a few hours, a few days, a few months, but they don't persist in that particular location due to competition that's there, or because there's defense cells or chemical changes, body changes that prevent them from remaining long term. Here's just a list of some of the different resident microbiota that we do have. This is not so that you can memorize all the different microbiota, but this is just to show you the variety of microbes that do live typically within the human body or on the surface of the human body. Since we have a lot of normal microbiota, it has to come from somewhere. So most of the normal microbiota is going to come into contact with us very early on during our life. So initially, these microbiota are going to develop from microorganisms that contact us very shortly after birth or even during the birth process. And so most microbiota that we do have, they're going to be established during the first month of life. So that's going to be our resident microbiota, those permanent members. And then there are certain things that can happen that can cause that normal microbiota to become opportunistic pathogens. So this would cause them to become pathogenic or be involved in some type of parasitic relationship. And things that can happen that would cause that would, first of all, be introduction of a member of the normal microbiota into an unusual location in our body. We could also have immune suppression, so our immune systems may not be able to combat those normal microbiota and keep them in kind of check. There could also be changes in the normal microbiota that disrupt the overall balance. And if that happens, we could see an overabundance of one of those organisms that might normally not be quite so prevalent. When we start talking about infectious diseases in humans, those infectious diseases or those pathogens, they have to come from somewhere. So these are going to be what we call reservoirs. Reservoirs are sites where pathogens are maintained as a source of infection. Some animals act as reservoirs, so we will have the pathogens established in those animals. They're able to grow and reproduce in those animals. And then if we come into contact with the animals that have the pathogens in them, we can become infected. Zoonoses are going to be different pathogens that can reside in an animal host, so the animal can serve as a pathogen. And humans can become infected by these in a number of ways. So if we come in contact with that particular animal, we could get infected. If we come into contact with the animal's waste or with different animal products, so with their fur, we can eat the animals and become infected. We could also become infected through arthropod transmission. And what that's referring to is if you have something like a tick or a mosquito that feeds on that particular animal and then it does feed on us, then it can transmit that pathogen to us. Now, typically, the more similar the animal is to humans, the more likely it is going to be that it can act as a reservoir for that particular pathogen. This table right here is showing us some common zoonoses. There are a number of them, and if you look at the animal reservoirs here, you can see that they're all pretty much mammals that we're looking at. And again, if the animal is more similar to humans, then it's probably going to be much more likely to be able to serve as some type of pathogen for a virus or a bacteria, a fungi that's going to be able to infect human beings. We can also have humans act as reservoirs for infectious diseases. Some of these are going to be individuals that are not yet showing any symptoms, so perhaps later the disease will develop and they will show symptoms. And in some cases, the carriers are not going to have any effects at all. They will just be a reservoir where that particular pathogen is going to be able to multiply and later spread to other individuals. Here's just an infographic that shows one of the problems with tuberculosis. TB is tuberculosis, and that is something that does reside in humans. Humans act as a reservoir, and this is a big challenge across the world. There are, you can see that there are more than 2 billion people, or a quarter of the world's population, that are infected with TB, and we have a lot of deaths yearly that result from that particular infection. So in this case, you do have a number of carriers that do not actually exhibit symptoms, and so if they are coming in contact with other individuals, they can spread that disease to those other individuals. Non-living reservoirs can also be a problem. A lot of times we're talking about things like drinking water, which is what we're seeing in this picture here. Cholera resides in drinking water, unclean sources in many cases. This could be dirty linens, this could be clothing, shopping carts. Those would be the non-living reservoirs that we're talking about. So there are a number of reservoirs around us that do contain those pathogens that can potentially infect us. If we talk now about the steps that will be involved in basically getting an actual infection inside of a host, the first step is that there has to be some kind of an exposure of the host to the microbes from that reservoir. So that's going to be exposure to the animal reservoir. That might be exposure to a human carrier. It might be drinking some unclean water that actually holds that particular pathogen. But in any case, we're exposed, and then there has to be some type of entry of that microbe into the host organism. Once that happens, if that particular pathogen is going to cause an infection or an infectious disease, it needs to become established in the host. The first step to infection is going to be contamination. This is where we are exposed to the microbes from that reservoir source. And so contamination, by definition, is presence of microbes in or on the body. The results of that contamination can vary quite a bit. So possible consequences or possible results of contamination: first off, the microbes might remain where they first contacted the body, and they may not end up causing any harm at all. If they remain there and they're not causing any harm, then they're probably just going to become part of our normal or permanent microbiota. Another possibility is that the microbes remain on the body only a short time without causing any harm, so they stay for a while and then they leave. In that case, we would call them transient microbiota. And then the other possibility is that we could get the infection. So the microbes, not only do they remain, they overcome the body's external defenses, they're able to multiply, and they do become established. That is what we refer to as infection. So contamination is the first step. Once we have contamination, we have to have the entry of the microbe into the host if it's going to ultimately cause infection. We have a number of places where the microbe can actually enter our bodies. We call these portals of entry, and you can see them on this diagram here. They're pretty much all of these openings or orifices that we do have. The mouth, the nose, we have the vagina. We can also have what are not natural entry sites, things like broken skin or insect bites. So if we look a little closer at these portals of entry, the skin, this one does have a lot of natural openings that we might not normally initially think of, but we do have, you can see it in the slide here, there's entries or pores where the hairs are, the sweat glands. Any of those, if there's microbes on the surface, there is this potential that they could actually enter the body. So that's what we're talking about in this second step, is that we've come into contact with these microbes, there's been contamination, and then they're able to enter our body. They can certainly enter through our skin without any broken surfaces or injuries happen to be there. Another place where they commonly enter is going to be mucous membranes. Mucous membranes do actually line all body cavities that happen to be open to the outside world. So there are a lot of mucous membranes. And then the placenta is also one that maybe we don't think of so much, but the placenta is the connection between the mother and the fetus. And so if the mother has something, it can be transmitted to the infant or to the fetus through the placenta. And then the last one is going to be the parenteral route. This is really circumventing the normal portals of entry. And when we're talking about circumventing those normal ones, this would be something like a cut. It's not a natural opening, but it is something that can allow for a microbe to actually get into our body and then later potentially lead to an infection. The last step is going to be the actual establishment of the microbe in the host. For establishment to occur, there has to be the adhesion process, which is attachment of the microorganism to cells. Now, a lot of microbes have specialized structures that allow them to actually attach to the cells of their host. This is going to require the use of adhesion factors, which are special proteins, and it may determine the specificity of the pathogen and the host. So not all pathogens are capable of infecting all hosts, and this might be the reason why some of them don't infect a particular organism, because there might not be a match between the adhesion factor and some type of structure in the host cells. You can see in this picture right here that we do have these little structures on the outside of our microbe. In this case, these are acting as adhesion factors, and you can see how they're actually binding and linking up with this sugar molecule that's on the host cell. So that's what we're talking about with adhesion factors. They're going to allow the host to actually match up, and the microbe is going to be able to stick. Disease is going to be an infection severe enough to interfere with the normal functioning of the body. This is really going to cause a change from a state of health. And anytime we have some type of change from a healthy state, we do call that morbidity. So disease can cause morbidity, a change from our healthy state. And there's different manifestations of disease. So different ways that we can actually see the effects of this. And this is really in symptoms and signs. Symptoms and signs are a little bit different from each other. Sometimes they're very related to each other. But when we describe symptoms, this is a subjective characteristic of a disease. And this is something that's only felt by the patient alone. This might be something like nausea. You might feel nauseous, but nobody else can really feel the nausea. They can't say really whether you're nauseous or not. Dizzy might be another one. So dizziness is something that just the patient can actually feel. The signs are going to be the observable characteristics of the disease. And these are things that other people can actually observe. They can measure them in many cases. This could be a health professional. This could be a friend, somebody that's there with you. A fever is something that can be measured. You can definitely use a thermometer and you can tell if somebody has a fever. We can see vomiting. That is a sign that you do feel nauseous. So those are the differences between signs and symptoms. Again, a lot of times they are tied together, they're correlated. You have a symptom of nausea, but you have a sign of vomiting. Here's a table that just lists symptoms and some signs, just some variety that we do have there. Itching would be another example of a symptom. Itching is only something that the patient can feel. Scratching, that's going to be something that is an actual sign. A syndrome is going to be a group of symptoms and signs that kind of go together, and they collectively characterize a particular disease, a particular abnormal condition. So we do talk about a lot of syndromes, and then we say, okay, these are characteristics of that particular syndrome. There are a lot of different causes of disease. We focus in this class really on the ones that are caused by pathogens or microbes, but those are not the only types of diseases that we can have. Here's a list of some different causes of disease. There are certainly a lot of hereditary disorders or diseases. These are things that are carried in our DNA. There are congenital diseases. These are things that we are born with, which might be caused by heredity. It might be caused by our environment in the womb. And then you can see a number of other causes of disease listed in this table. So it's definitely important that we understand that every single disease that we might obtain or we might actually exhibit symptoms of and signs of, this is not necessarily always going to be the case of some pathogen infecting our body. Now, Robert Koch, he did do a lot of research on pathogens and trying to establish ways to determine whether or not a pathogen is responsible for causing a particular disorder or disease. And he established some steps which we recognize today as being very effective steps in proving whether or not a pathogen is responsible for causing a disease. And these are called Koch's postulates. It's a set of essential conditions that must be demonstrated in order to prove that a particular microbe is pathogenic and it is responsible for causing a particular disease. Now, if we go through the steps of Koch's postulates, the first one is that number one, the suspected agent, whatever that happens to be, whether it's a bacterium, a virus, a fungi, it has to be present in every single case of the disease that is observed. So if that condition is met, we can continue to look at it as a potential cause of disease. If it's not found in every case, then that's going to rule it out as the cause of that disease. The second one is that the agent must be able to be isolated and grown in pure culture. So that would be something that we could do in a laboratory. Number three, the cultured agent must cause the disease when it is inoculated into a healthy, susceptible experimental host. So basically we grow up a culture of that particular microbe and then we want to reintroduce it and see if it is going to cause the disease. And then lastly, the same agent must be re-isolated from the diseased experimental host. So again, just to sum it up, every single individual that we examine that has the disease should have the potential pathogen inside of them. We want to be able to isolate that pathogen, grow it in a pure culture. Then we want to take the pathogen, we want to introduce it into a new host. And when we introduce it into that new host, we expect it to cause the disease again. And then after it causes the disease again, and we see the signs and symptoms of that, we re-isolate that pathogen. If we can fulfill all those steps, then we can say that yes, that particular pathogen is responsible for causing that particular disease. Even when we follow all of those steps, there are some cases where we're just not able to conclusively prove that the pathogen is responsible for causing that disorder because we're not able to go through all the steps. So there are some exceptions here. One of them has to do with the fact that some pathogens cannot be cultured inside the lab. They just do not grow under typical lab conditions. Another problem is that some diseases are caused by a combination of pathogens or by a combination of a pathogen and then also a physical, environmental, or even a genetic cofactor. So that's very hard to replicate that particular set of conditions. And then lastly, and this is really an important one when we're talking about human diseases, is that there are a lot of ethical considerations to consider. So this might be that we don't want to take an individual and use them as an experimental host and infect them with that particular isolated pathogen, because of course we don't want to knowingly infect people just for the sake of proving the cause of a disease. So all of these do represent a bit of a problem when we try to follow Koch's postulates very, very strictly.